हेलो फ्रेंड्स, वेलकम बैक टू माय यूट्यूब चैनल। इस वीडियो में हम एमएससी जूलॉजी फोर्थ सेमेस्टर का सब्जेक्ट बायोकेमिस्ट्री का केमिकल बॉन्ड एंड सेकेंडरी बॉन्ड का स्टडी करेंगे, जिसका इंट्रोडक्शन है। केमिकल बॉन्ड प्ले ए वाइटल रोल इन बायोकेमिस्ट्री, इनेबलिंग द फॉर्मेशन ऑफ मॉलिक्यूल नेसेसरी फॉर लाइफ. These bonds hold atoms together within molecules and determine the structure and function of biological macromolecules like protein, carbohydrates, lipid, and nucleic acids. In the context of protein, understanding chemical bonds is essential for comprehend their structure, function, and interaction. This essay focuses on peptide bonds, secondary bonds such as disulfide hydrogen or nonpolar, and ionic bonds, highlighting their characteristics and importance in biochemistry. Definitions of biochemical bond. A chemical bond is an attractive force that holds atoms or ions together to form molecules. These bonds can be classified into two main types. First, primary bond. Second, secondary bond. Primary bond, it is a strong bond such as covalent and ionic bonds that create stable compounds. Second, secondary bond, its weaker interactions such as hydrogen bonds, van der Waals forces, and hydrophobic interactions, crucial for maintaining the three-dimensional structure of biomolecules. Peptide bonds. Definitions and formations. A peptide bond is a covalent bond formed between the carboxyl group of one amino acid and the amino group of another through the dehydration reactions. This bond is the backbone of protein structure, linking amino acids into polypeptide chains. Characteristics. First, strong and stable, resisting hydrolysis under physiological conditions. Second, planar in nature due to partial double bond character, limiting rotations and giving the protein a rigid structure. Third, essential for protein synthesis as it defines the primary structure of proteins. Biological importance. Peptide bonds form the linear sequence of proteins, determining the primary structure. This sequence ultimately governs the protein's folding and functions. Secondary bonds in biochemistry. Secondary bonds, although weaker than primary bonds, are crucial for maintaining the shape and stability of biomolecules like protein and nucleic acids. First, disulfide bond. Definition and formation. Disulfide bond is a covalent bond formed between the sulfur atom of two cysteine residues. This linkage occurs through an oxidation reaction. Characteristics. First, stabilize the tertiary and quaternary structure of proteins. Second, commonly found in extracellular protein like insulin and antibodies. Biological importance. Disulfide bonds provide structural stability to protein, ensuring they retain their functional conformation in varying environmental conditions. Second, hydrogen bonds. Definition and formation. A hydrogen bond is occur when hydrogen atom covalently bond to an electronegative atom, example oxygen or nitrogen, interact with another electronegative atom. Characteristics. Weaker than covalent bond but critical for molecular interactions. Second, directional, contributing to specific protein folding and DNA double helix stability. Biological importance. Hydrogen bonds stabilize the secondary structure of proteins, alpha helix and beta sheets, and maintain the double helix structure of DNA. Third, non-polar of hydrophobic interactions, definitions and formations. Hydrophobic interactions arise when non-polar molecules or region of molecules aggregate to minimize contact with water. Characteristics. First, not true bonds but are critical for stabilizing protein folding. Second, drive the formation of lipid bilayer in cell membranes. Biological importance. Hydrophobic interactions ensure the proper folding of proteins and the formation of membrane structure essential for cellular function. Fourth, ionic or electrostatics bond. Definition and formation. Ionic bonds occur between oppositely charged ions such as positively charged amino groups and negatively charged carboxyl group. Characteristics. First, stronger than hydrogen bond but weaker than covalent bonds. Second, sensitive to changes in pH and ionic strength. Biological importance. Ionic bond stabilize the tertiary structure of proteins and facilitate interaction between enzyme and substrates. Characteristics of chemical bonds. First, strength directionally. Third, reversibility. First, strength. Peptide bonds are strong, while secondary bonds are varying in strength. Second, directionality. Bond like hydrogen bonds exhibit specificity in orientations. Third, reversibility. Secondary bonds are reversible, crucial for dynamic molecular interactions. Fourth, stability. Disulfide bond provide stability under stress. Fifth, flexibility. Secondary bonds allow biomolecule to adapt to functional requirement. Applications in biochemistry. Protein folding. First, secondary bond guide the folding of polypeptides into functional protein. Second, enzyme activity. Ionic and hydrogen bonds facilitate enzyme substrate binding. Third, cell membrane. Hydrophobic interactions drive lipid bilayer formation. Fourth, DNA stability. Hydrogen bond maintain the double helical structure of DNA. Conclusion. Chemical bond ranging from strong covalent peptide bonds to weaker secondary bonds are fundamental to biochemistry. They define the structure, stability, and function of biomolecules, enabling life processes. Understanding this bond provides insight into protein folding, enzymatic activity, and molecular interactions, which are pivotal for advancement in biotechnology and medicine. References.